Okay, so you guys can hang on to your exams until the end of class, and then I'll recollect them again. We're almost done with the meiosis lecture here. Make sure you guys can see. Okay. So this slide is hopefully mostly a review. Yeah, so I talked about, and I kind of harped on where genetic diversity is introduced during meiosis. So that's the really interesting and I guess exciting part of meiosis, is that you mix up the genes. So in mitosis, you're creating clones of cells, right? You're just creating the same genetic cell genetically over and over again. In meiosis, you get this whole new combination of traits. So the two spots where this happens, and you definitely want to know these, are prophase 1 and metaphase 1. So that first round, before we've even split that one cell that we started with into two, you get a lot of the genetic variability introduced. So crossing over is one example of where that can happen, and that is kind of diagrammed over here on the right, where you have the paternal and the maternal DNA essentially pairing up and then switching sections. So that's what this, obviously it's aptly named crossing over, and they actually switch segments of their chromatids with each other. Those sections have the same genes on them, but they might have different versions of those genes. They might have the same versions of those genes. It's just completely random. There's no rhyme or reason to where the crossing over actually happens. So crossing over is one big way that genetic diversity is introduced during prophase 1. And then independent assortment is the second one, which isn't quite as straightforward maybe, but this happens in metaphase 1. So we'll imagine that we've already gone through, obviously already through prophase 1. So we're into the next step. The tetrads, these combinations of chromosomes, the paternal and the maternal, are lining up along the midline. Metaphase, they're lining up along the midline in basically two lines next to each other. Depending on which way they're arranged, which is on the left and which is on the right, determines which set of DNA goes into each eventual cell. So here I have possibility 1 and possibility 2. Both of these are equally probable. So here we have well, it's blue on there and kind of green on here, but the bluish green chromosome is on, let's see, I guess we're looking at the little ones. So the bluish green chromosome in possibility 1, the little one, is on the left. Possibility 2, it's on the right. Again, that's totally random. There's no rhyme or reason as to how they line up. Completely random. That's how we get this variation. So you guys on Zoom, the blue here is on the left, the little chromosome. And possibility 2, it's on the right. So just switches the position, which, as you imagine, when that cell splits in two of the chromosomes are going to one cell, 2 are going to the other cell, and they're splitting down the middle, right? So in the end, you'll have different combinations of chromosomes when we get to metaphase 2, and then eventually to the gametes, based on the fact that these just originally lined up differently from each other. So in possibility 1, you have blue or green, in this case, all in one cell. Possibility 2, you have the green and the red, or the blue and the red, if you're on Zoom. So you can see at the bottom what that does to the 4 gametes that are produced. So they're all going to be, you're going to end up with 2 different combinations of gametes. Any questions about that? Genetic diversity stuff. Okay. Meiosis is not perfect. I think that's where I cut off the Amoeba Sisters video and said I'll talk about this on Friday. there's a number of errors that can occur with, in a lot of different aspects of the cell cycle. When we're talking about meiosis, specifically, there can be issues with the chromosomes not separating correctly. So if we think about, we'll just imagine this cell basically only has one chromosome, 2 copies of it. If for some reason, normally one chromosome is going to go into each eventual cell after the first round of meiosis, right? So they should separate evenly between the next round of cell division. Sometimes that doesn't happen. So you have this, what is called non-disjunction. So the chromosomes don't separate. So one cell gets both of them, which is too much DNA in one cell. The other cell gets none. So they don't, non-disjunction. They don't move to opposite poles during anaphase, either 1 or 2. So anaphase 1 or 2. This can happen when you have the homologous chromosomes still, tetrads, or it can happen when you're splitting those 2 cells in the second round of meiosis into 4. So for instance, even if you get through the first round of meiosis just fine and each cell gets a chromosome, awesome, then maybe one of those cells goes on and non-disjunction happens in the second round of meiosis. So each of these chromatids should go into each cell, kind of like this. But they don't. They end up in the same cell. So now you have, again, too much DNA and no DNA. Both of those are major problems. So for you guys on Zoom, if you couldn't see what I was pointing at, if you go from meiosis 1 to meiosis 2, this can happen as well. The chromatids just don't separate, which they should, like they do here in the yellow. So this creates what we call aneuploid gametes. So that's gametes that just don't have the right number of chromosomes. They can have too many or they can have too few. So non-disjunction leads to aneuploid gametes. And this is actually one of the most common causes of kind of spontaneous abortion in humans. So when the egg just doesn't form correctly, or either the egg or the sperm didn't form correctly and doesn't have the right amount of DNA in it. When it goes to fertilize, either the egg or the sperm, when the egg and sperm come together, they don't have the correct number of chromosomes, so the zygote really never gets going. And we'll talk more about some genetic disorders next week, probably, maybe the week, I think next week. I should get into them at least by Wednesday next week. So this is just one kind of generalized example of what can happen when the genetics don't go quite right. Okay. Oh yeah, and then this final slide, kind of just for your info, I'm not going to ask you to compare and contrast meiosis and mitosis, but this can be helpful if you're getting a little bit confused, or if you understand mitosis really well and you're trying to kind of figure out how meiosis fits into that. This gives a good breakdown of the main pieces. So meiosis, again, it's making distinct gametes. So they're all unique. They're not clones of each other. Mitosis are just cloning body cells. So these are in reproductive cells. Meiosis happens in reproductive cells. Mitosis happens in the rest of your body cells. And we're moving from a diploid state at the beginning of meiosis to a haploid. In mitosis, we're staying diploid the whole time. So usually mitosis and meiosis are taught really closely to each other, but for some reason in our schedule, we decided to kind of separate them. It makes sense because we're getting into genetics, and meiosis has a lot to do with genetics. So anyway, like I said, I'm not going to ask you to compare and contrast, but just kind of for your knowledge, the differences between meiosis and mitosis. the main ones anyway. And that wraps up, I think, our meiosis chapter. Yay. All right. Here we go. So now we get into what I think is some of the most interesting stuff in general bio. Sorry, I can't do two things at once. Hang on. There we go. Okay, so you guys should be able to see the intro to genetics slides now on Zoom. So yeah, genetics are fascinating. It's just really interesting to think about how many different things have to happen to make us who we are. And that's sort of what we're going to be talking about. We're going to start with some basics, obviously, and then I'll get into some more kind of human-centered stuff. But we sort of have to step back and talk about where genetics, where the whole process or the study of genetics kind of started. So we're going to go back in history a little bit, and then we'll talk more about some human-related and also non-human related genetic stuff. So this is where we get into Punnett squares. If you guys remember Punnett squares from high school, those are always fun and, well, I guess fun for biology and pretty straightforward. So it's fun to figure out kind of what the ratios are, the offspring of two individuals. Okay, enough of that. Into genetics. So first, I talked about some of it. So I talked about homologous chromosomes. I'm going to go into a little bit of detail on genetics terms here over the next few slides. Well, I guess it's spread apart across a couple different sections, but there's going to be a good bit of terminology that we have to get through in order to talk about genetics. So these are all terms that you'll hear a lot going forward. So we know generally what a chromosome is, right? It's a bunch of DNA condensed. So on these chromosomes, you have genes. So a gene is simply a part of a chromosome. And I'll give you a more detailed explanation or definition of a gene in a few slides. But it's just a part of a chromosome that codes for some kind of character. So it can code for flower color, for example. We're going to talk a lot about pea plants. So we'll talk about peas and their flowers, because that's where all of this kind of started. So we're going to do a little bit of plant stuff, which we haven't done much of. Okay, so genes are just a part of a chromosome that codes for a specific character. The allele, like I defined last time, I think, is just the alternate versions of that gene. So on these two chromosomes, these are going to be homologous chromosomes. They have the same genes. And here they have a flower color gene on both of them. However, this chromosome, the blue one, has the allele for purple flowers, which is denoted as big P. I'll talk about the big and little in a minute. This chromosome has also the same gene for flower color, but it has the allele for white flowers, little p. So for you guys on Zoom, the blue chromosome has a different allele than the red chromosome, but same gene, different alleles. The locus is just the location of a gene on that chromosome. Yes?
Is the allele like your recessive trait?
The allele can be dominant or recessive, yes. Yeah, and we'll talk a lot about dominance and recessiveness. So here, the big P indicates that this allele is dominant over this allele, the recessive, yep. Okay, so locus, just location. I won't use that term, I don't think all that much, but it might pop up here and there. Gene and allele you'll hear over and over and over and over and over again. All right, so some history here. Good old Gregor Mendel. So you may have heard Mendel's name. I think this chapter is called Mendelian Genetics because it's all based on his work. So he's the father of modern genetics. He was a monk in Europe. Was it Switzerland? I can't remember off the top of my head, somewhere. a monk in Europe who liked to grow peas. And he was around in the 1800s, so like mid 1800s. And his work with pea plants actually is what led to our understanding of modern genetics, even though he had no idea what a chromosome was. He didn't know what a chromosome was. He just crossed all these pea plants and kind of saw what happened and figured out the basics of genetics from that. Here we go. So that's the next bullet. I got ahead of myself. So he developed the basic understanding of how traits are passed down through generations. I'm not sure what originally interested him in peas in particular, but it was convenient. They're convenient to deal with because they're really easy to deal with. First off, they grow really fast and they have a lot of offspring. And plants are so much easier to control than animals. You can control exactly what plant mates with another plant because you just take a little paintbrush and move pollen from one plant to another plant. You don't have to worry about any other kind of unanticipated mating. You can cover up the flower of one plant to make sure no other pollen actually gets in there. So you can control pollination so you know exactly what plant is mating with what plant. And they also have easy to identify traits. So these pea plants in particular, he looked a lot at, let me bring this up. He looked at flower color, seed shape. All of these things are easy to see with the naked eye. You don't need a microscope. You don't need any kind of special equipment. You can just, you can easily determine these different traits. So he also looked at the shape of the pod, the color of the pod, the pea pod, where the flowers were, how long the stem was. He looked at all kinds of different stuff. You don't need to know everything from here. This is just kind of, we'll be talking about some of these traits, mostly flower color, and seed shape. So these first three we'll talk about a good bit. But yeah, peas are just easy to work with. So it worked out well that he was for some reason interested in them in the first place. Okay, a few more terms here. So I mentioned character, I already defined that. Or did I define it? No, I mentioned it in a gene. So a character is the heritable feature. And when I say heritable, it just means it can be inherited from one generation to the next. And it's going to vary among individuals. So from the previous slide, the examples would be like flower color, seed shape. That's a character. The trait are the options for that character. So purple flowers, wrinkled seeds, or wrinkled seeds, smooth seeds, etc. So character and trait, those are often kind of confused. So I'll try to make sure I use those as accurately as possible. The character is the feature, the trait is the option for that feature, the variant. Okay, more terms here. All right, so a more detailed definition of gene. So we know it's the unit of hereditary information for some kind of character. It's based on, and this goes back to those biomolecules that we talked about a long time ago, based on specific nucleotide sequences. Okay, so it's essentially a code for a trait. If you remember, like DNA is made up of a bunch of those nucleotides. So the individual pieces that make up DNA or a chromosome are just a bunch of nucleotides strung together. I think I mentioned the nucleotides like thymine, cytosine. There's 4 of them for DNA and 4 for RNA. So you can think of it as a code. It's just a code that determines what trait that gene or that character is going to show. So these are just some simple examples of some genes: eye color, blood type. What the heck does that say? Hair color, hair color, growth. Yeah, so like height, I guess. So those would be genes, and you can just think of genes as these segments of chromosomes. And if you pull out, remember, a chromosome is just really condensed DNA. So we have that double helix of DNA. A gene would be a section of that. So a bunch of nucleotides strung together. Alleles are, like I already said, an alternative version of a gene. So this is where we get into the dominant and recessive aspects. You can have, and in the most simple of scenarios, you have a dominant allele and a recessive allele. The dominant allele is always expressed, so you always see it. It's usually denoted by a capital letter like big A or big P, like I showed you earlier. The recessive allele, it's only expressed if there's no dominant allele present. And I'll show you what that looks like on the next slide, basically. These are denoted by a lowercase letter. So if for one gene, you're going to use the same letter for dominant or recessive. So that's why for the flower color, big P was purple, little p was white. We don't use little w. That would indicate that it's a totally different gene. So you use the same letter. So big P, big A, little a, etc. For our purposes, until we get into some of the kind of complicated or the exceptions, I guess, we're going to have 2 alleles for every gene. There is a scenario where there's more than 2, but we'll talk about that later. So 1 from the mother, 1 from the father. Okay. And a couple terms you may have heard also when dealing with Punnett squares are homozygote and heterozygote. So first, a homozygote is an individual with the same allele for some gene. So if we have whatever gene B is, well, in this case, it looks like it's flower color. The homozygous individuals are going to be either big B, or little b, of the same. So these are our 2 homozygotes. You can be, I think I have this later, homozygous dominant or homozygous recessive, depending on whether the plant has the little b or the big B allele. But either way, it's the same one. They don't have a mixture. They don't have a big B and a little b. That's going to be considered a heterozygote. So a heterozygote has 1 dominant and 1 recessive. And the last couple of terms for this part are genotype and phenotype. The genotype is the genetic makeup of an organism. So what alleles does it have? Does it have 2 big B's, or does it have a big B and a little b, or does it have 2 little b's? That's the genotype. The phenotype is just what does it look like? It's the physical expression of the genotype. Its physical appearance. So the genotype of this flower, the big B, is going to be big B. So genotype is big B. Phenotype is purple. So phenotype is what we see, essentially. Genotype is sort of the underlying code that tells this flower what color to be. So you want to be sure you can tell me what a genotype or a phenotype, given that information, of an individual would be. So what is the genotype of this one in the middle? What would you say the genotype is? If the genotype of this one is big B, what's the genotype of this one?
Big B.
Yeah, big B, little b, thank you. I think everyone knew that. You just want to know the answer. So the genotype for this heterozygous individual is going to be big B, little b, and what's the flower color? Or what's the phenotype, sorry? Purple, yeah. Because that big B is there, it's automatically going to be purple. Over here, our genotype is little b, right? There's no dominant allele, so if it's all recessive, that means the phenotype is going to be recessive as well. So the flower will be white. So the phenotype is white, purple, purple, and then these are the genotypes, the B combinations. Okay. And we're going to do some crosses. We won't do them today. We'll get into them on Monday. When we do crosses, we're going to talk about parental generations and then the offspring generations. So these are just a few, these are very straightforward. The last three terms are very straightforward. So they're just different names for different generations. So the P generation is going to be what you start with. If you start with a tall plant and a short plant, those are the parents, the parental generation, the starting point. The offspring of two P generation individuals crossing is going to be the F1 generation. So two P generations crossing, you get F1. If you cross two F1 individuals, you get F2 and so on. You can keep going. We're not really going to go beyond F2. And in this case, plants are weird. They can self-fertilize, so they don't even need another individual. So P, F1, and F2. And with Punnett squares, we're going to look at the genotype and phenotypes, the genotypes and phenotypes of what these crosses produce. So we'll look at what the parents are and then figure out what the F1 generation, what the options are for the F1 generation, what the chances are that an offspring would be homozygous dominant versus heterozygous versus homozygous recessive. And then we'll do the same into the F2 generation. Okay, so let's see. While we're talking about meiosis, this is probably, or we just got done with meiosis, so I'm going to get through a couple of Mendel's laws here, and then we'll be done for the day. I don't want to get into Punnett squares because I don't know that we'll have enough time to really talk about them. So when we think about the traits that offspring have, there's a couple of laws that Mendel essentially came up with that helps us understand what's going on. So the first is going to be the law of segregation. And this is where two alleles for a particular character, so we're dealing with alleles here, two alleles for a certain character are going to separate from each other as gametes are formed. So they're separating from each other because they are on homologous chromosomes which separate, and then the chromatids separate as well. So the alleles segregate or separate from each other. This happens during anaphase, which is where the chromosomes are separating or the chromatids are separating from each other. So during 1 and 2. So the first round of meiosis and the second round. That means every single gamete at the end, because we only have one copy of the DNA at the very end of meiosis, each gamete has only one copy of that allele. So let's say, yeah, we had 2 big A's and 2 little a's. All 4 of those are going to be split across all 4 gametes, assuming everything goes as planned. There's no non-disjunction or anything like that. So assuming meiosis happens correctly, you end up with one of the alleles in each of the gametes. So that's the law of segregation. Alleles are going to separate. And then the law of independent assortment deals with genes on different chromosomes. So these are not the same gene. So when we were dealing with the alleles, those were the same genes, right? They were just different versions of the gene that separated out. Now we're talking about totally separate genes on separate chromosomes. So we'll say hair color and eye color. Those are going to separate from each other randomly as well. So 2 or more genes on different chromosomes assort independently or separate from each other randomly during gamete formation. An easier way to think of this, I think, because the definition doesn't always hit home for me, the traits, or the characters. The characters, the genes, are not linked to each other. It doesn't mean because you have brown hair, you can't have blue eyes, that kind of thing. So they're not linked. They're not inextricably linked to each other. They can be separated. You often see combinations that are similar. So if you have darker hair, you usually have darker eyes, etc. But they're not, in general, they're not linked to each other. We'll talk about some exceptions to that. But that is the law of independent assortment. And this occurs during metaphase 1. That's where those chromosomes are lining up and they can kind of separate, line up in multiple different ways along that midline, which means the chromosomes, those genes are separating in totally random ways if they're not on the same chromosome. Okay, so those are his 2 big laws that you want to know. Yeah, and we'll stop there for today. It's Friday. I think that's plenty. We'll get into Punnett squares on Monday and talk about what Punnett squares show and how to interpret them. And I'll take y'all's exams back on your way out. And have a good weekend.